9.1, the section that never quits. Just going back real quickly. This whole chapter is about what do you do and how do you deal with breaking bonds. When you break bonds, here, we got two situations that are happening. One is this rearrangement of the atoms into a more stable state. So you're going to rearrange the atoms from that original molecule into something else that's more stable and capture that little bit of energy that comes from that change into either the ability to drive processes that need energy or save it as ATP to do real work in the cell. The reason being, this is a spendable amount of energy, whereas the electrons that are generated as you break bonds, way too energetic. And so there has to be a sequential, stepwise way to dab a toothpaste, squeeze the energy out of the electrons. So for this question, we're starting to make some connections between things we've already learned. Explain the importance of oxygen in redox reactions. So this passing electrons back and forth. Something gains, reduced. Something loses, oxidized. So what's the importance of oxygen in that gain-loss of electrons reactions? Focus your explanation on oxygen's properties and how those relate to cellular work. Okay? So let's start off with, we've got like a mantra. Water is polar, right? Behavior is a response to a stimulus. What was the other one we had that I said? Now we've got another one. Oxygen is highly electronegative. That's what we want to start with. So that's where I'm going to go. So this is 8. So oxygen. Now I can't get that out of my head. What the other one was. Oh, enzymes or proteins. Okay, so I'm going to write that so I don't forget. Water is polar. Whoops. Behavior is a response to a stimulus. Enzymes are proteins. So now we've got like 4 that we've got. Oxygen is highly, what in the world, electronegative. Wow. Okay. Oxygen is highly electronegative. What does electronegative mean? It means that, let's go over here and look at this one word I say. It's kind of crazy. An oxidizing agent, isn't that interesting? Wonder why it's called oxidizing. Causes something else to be oxidized and becomes reduced itself. And we interpreted that correctly to mean, hey, I steal electrons. Oh, why would we name that after oxygen? Because oxygen is highly electronegative, which means that it can pull electrons to itself. So because it has this high electronegativity, so therefore it pulls electrons away from, steals other molecules. We'll go with that. It's an oxidizing agent. It's a bully. It snatches away electrons. It really holds electrons tight. So it holds electrons tight. Tight, tight. How does that relate to cellular work? Well, when you think about cellular work, you need to think about this making ATP for cells to do work. That's what it's really talking about. How is oxygen part of these redox reactions? And we need to focus on particularly oxygen's role in making ATP for cells to do work. And we have to think about its role is going to be electron-based, because that's what it's good at, stealing electrons away. So that must mean that it's going to have something to do with this passaging, transporting of electrons. So we're just going to draw the process right here again. So we're going to start back at glucose. So we're going to repeat what we draw several times. Hopefully we'll get better at it. So if we start with glucose, and we're going to break bonds. So we're going to be a little bit more detailed as we go. So we're breaking these carbon-hydrogen bonds. And glucose has 6 carbons, right? But no, we're not breaking them all like poof. We're going to break 1 carbon off at a time. So it's part of the pathway. It's sequential. Every time we break a bond, you generate 2 hydrogens, 2 electrons per bond. One of the hydrogens becomes part of the gang where that we create a proton gradient. So 1 is used to create a proton gradient. And then we have 1 hydrogen now and 2 electrons that we can't let go crazy because glucose was oxidized and lost these electrons. So something has to take them. They're given to NAD, empty bus, oxidized form, gains these 2 electrons and that hydrogen ion and becomes reduced, full bus, to form NADH. That's the same story we had before. Where's oxygen in this? Like, where are you going, woman? We didn't talk about now where NADH goes. It's an electron carrier. So where are you going to carry things? Well, let's let it be carried to the processing plant for electrons. Because it's got 2 on the bus, it's going to drive the bus, electrons on the bus, to the electron transport chain. So let's write that. So NADH, with its 2 electrons, carries the electrons and that hydrogen, little groupie, to the processing plant for electrons, which is the electron transport, which makes sense because we're going to pass them back and forth chain. So the ETC. So now we're going to put together a whole bunch of stuff that we've done before. So this electron transport chain, let's just picture what it has to be in our head. There's going to be a series of electron acceptors that are going to gain that electron, so they're being reduced, pass it to the next acceptor, that means they're oxidized, the next acceptor's reduced, and they're going to be this sequential passaging of that electron. That sounds like a perfect job for a membrane. So in the membrane, again, just like we did before, I'm not saying these are all saturated. I'm being lazy. We're going to have embedded in the membrane, we're going to have proteins that are the electron acceptors. So this is a protein built into the membrane. So let me put some more phospholipids. And then I'm going to have the next electron acceptor, again, which is a protein. And then, because that electron has a lot of energy, I'm going to have another electron acceptor that's a protein. So all of these are built into the membrane, which we learned before. When you put proteins in a membrane and you localize them, so this is localization, they can rely on each other to do their jobs sequentially. So the way this is going to work, here's your bus, NAD. I have 2 electrons. I'd like to drop them off, please. So it's going to drive over here. It has a parking space at this first protein. So NADH is going to give up its electrons to this protein. When it does, it becomes an empty bus. So it's going to be converted to NAD, the empty bus. So now NAD is regenerated. Hey, thanks for taking the electrons. See you in a little bit. And it's going to drive back to pick up more electrons. So it's going to keep picking up the electrons. Okay, picked up 2 electrons, full bus now, driving over to drop them off, dropping them off here. Thanks for taking my electrons. Going back to get some more. So you see this cyclic process. So we're going to simplify this because I told you we're going to add as we go. So we're going to let the electrons just be this blue highlighter. So when they get dropped off, they're going to be passaged from this protein that's now been reduced, right? It gained electrons. It's been reduced. It's going to passage the electrons to the next protein. So that's a redox reaction again. Now protein 2 is reduced. Protein 1 is oxidized, ready to pick up more. The electrons will be passaged again. Every time we do this, we're dropping the energy level of the electron. And so we're able to do work. What work? Well, thank you for asking. We will use that energy of the electron as it drops in energy to pump hydrogen ions from this side of this membrane over here against the gradient. As that electron is passaged to the next protein, that is going to be a drop in energy that will be used again to pump hydrogen ions from this side of the membrane where they're low over here where it's high, which requires energy. Thank you, electron, for giving me a little dab of toothpaste energy so we could pump some hydrogen ions. And we passage it again. Pump hydrogen ions against the gradient. Well, what happened to the electron? Did it get stuck at this protein? No, it did not. So after we have squeezed the life out of it, used its energy to pump hydrogen ions against the concentration gradient, this electron, now I'm making this simple, we'll come back and do more, is finally given to its final electron acceptor. So for every 2 electrons that you have run through here with 2 hydrogen ions, it's going to be given to, and this looks really weird as a half, it's going to be given to an oxygen. Oh, finally we're getting oxygen in our story. And we're going to create water. So ultimately, like I told you before, if you do what I said, which is, hey, you follow that electron, buddy. You follow that electron. Oh, okay, I'll follow it. It came out of glucose. Yes, it did. And it was given to a carrier. Oh, yeah, it was. It was put on the bus. And then it was driven over to the electron transport chain. And we whoop, squeezed it, whoop, squeezed it, whoop, squeezed it. And now it's calm enough to be given to oxygen as the final electron acceptor. And now we make water. So I told you this was the story of the serial killer electron that after it goes through its processing becomes mild enough to be Mr. Rogers and live next door and babysit your kids. So you make water. You couldn't have anything less harmful that you made. You made water. So now let's kind of put this in words. We've got this in words. It's over here. So now let's do the rest of this story, which now there's more to it, but this is a good part of it. So we're going to write the rest of it. So our story was, we'll just briefly write it. Glucose loses electrons when the bond is broken. Now it's one bond. We're doing this one at a time. So period. NAD gains, right? Gains the electrons. So we're going to just put is reduced. And becomes the reduced form that I've got 2 electrons on my bus, NADH, the full bus. NADH is a carrier, so it's going to drive those electrons to the electron transport chain. NADH brings the electrons to the electron transport chain and loses those electrons to this protein right here. So if it loses its electrons, now it's oxidized. It's going to be the empty bus again. Which is NAD, and goes back, it's a shuttle, to get new electrons. Okay? So that's the story of NAD and NADH. Now we have the story of the electrons. So the electrons are passed to a series of acceptors.
I didn't find acceptors on our
I didn't ask you to. A series of electron acceptors. Acceptor, acceptor, acceptor. Through, let's use our vocabulary, redox. If you gain, you're accepting the electron, you're in your reduced form, then you give up it in your oxidized form through redox reactions, each pass of the electron from one acceptor, right? Is this where I'm going with this? Yes, it is. From one acceptor to the next acceptor causes the electron to drop in energy. That energy, we're going to use it to do work because it's not a big amount. It's just a dab of toothpaste. That energy is used, here we go, to do cellular work. All right, finally I'm over here actually getting to the point of this. To do cellular work of pumping hydrogen ions, which we were generating them right there, remember? Against the gradient. So they're going from low to high. Okay? So that's the story of the electron. Now we're going to have, so after several passages, transports, redox reactions, after several handoffs of the electron through the acceptors. I don't know, I can't think about that right now. Through acceptors, the electron is in a low enough energy state to be given to oxygen. And oxygen is the final electron acceptor. What is that? It didn't look right. That didn't look right up here either. But is it? Okay. What do you think about that? So, we got the whole cellular work thing in there. We got oxygen's role in it. That's the story. Like if we can get this in our head, literally you have all the cellular respiration down pat except just tiny little details. That was 8. Let's look at 9. Explain the importance of the high number of hydrogen bonds found in fats and carbohydrates. Because remember, fats and carbohydrates are only, they're carbon, hydrogen, oxygen only. The difference is carbohydrates are lots of oxygen. Fats are almost no oxygen. Okay? So carbon has just had a lot of water really added to it, is what it is. Fats don't have that. So what explanation in the role of enzymes in the process of oxidation of these energy sources. Okay? So here's what they're wanting us to get. We've got this molecule that's got a lot of carbon bonds. So I'm just going to put ... to show it goes on. And in this case, we're going to look at like a saturated fatty acid. Because we've got all these single carbon bonds loaded with hydrogens. What happens is you have the ability to snap off a lot more than 6 carbons. Glucose has 6. You get 34 to 36 ATP by the time you get completely oxidized glucose. Fats have more energy because they have more carbon-hydrogen bonds. So that's what they're asking is the more carbon-hydrogen bonds, right, that you can break. Let's think about what that is. the more, what do you get when you break a bond? Electrons. More electrons to go to the electron transport chain, which we haven't even talked about what we use this proton gradient for, but you can guess it's going to be to make ATP, which equals more ATP. That's why fats have twice as many calories. Calories a measure of energy in food. So that's why they have twice as many as carbohydrates, because they have more carbons. So that's what the story of this is. More carbon-hydrogen bonds, more electrons go into the electron transport chain, and every time you passage an electron through there, you're building a gradient, which later will draw in how do you release the gradient to make ATP. Focus your explanation on the role of enzymes in the process of oxidation of these energy sources. What they're saying there is there's going to have to be some pathways to break these off. That's what they're saying. Enzymes, pathways are made up of enzymes. So let's just start with that. Pathways are really just little factories where enzymes are the workers. So they're made up of enzymes that each do one step of the pathway. So to fully oxidize a fat, lots of carbon. Like you could have 22 carbons, no problem. To fully oxidize a fat, so we're going to put here lots of carbon-hydrogen bonds, you're going to break them off one at a time. And that has to be done in a pathway with enzymes. So you do this one bond at a time. And you're going to use, so through an enzymatic pathway. That just means that all the steps have enzymes in it, because a lot of these processes would not naturally occur quickly enough, and so we use enzymes for it. 10. Explain the necessity of stepwise energy harvest in the oxidation, now we know what this means, breaking bonds, releasing, losing electrons, of fuel. So fuel would be things that are long hydrocarbons. Well, this is pretty simple. So that's 10. Let's make sure we understand. When they say oxidation, they mean breaking bonds. And the reason for this is because electrons are lost, so therefore there's this oxidation. So they're oxidized. That's where this comes from. So that's what they say when they say oxidation. They're saying fully breaking that molecule's bonds. When they say fuel, they mean carbon-hydrogen bonds, usually a lot. Okay? So what's the reason why stepwise is so important? Wow. Now we can answer that question. The reason, so stepwise, when they say stepwise, they mean one bond at a time. Don't just go snapping all 22 carbons apart in that long fatty acid, one at a time. So when you do stepwise, one bond at a time, then you don't have this. It's kind of the same thing like with this whole thing with COVID. People don't understand that that we're having the restrictions. It's not only for safety, because none of us have natural immunity to this thing. High likelihood we're all going to get it. We just don't need to all get it at once. If we all get it at once, we're going to overrun the hospitals, and we can't manage that kind of exodus of people out of houses into the hospital. It's the same thing here. When you break these one bond at a time, you have a sequential release of electrons instead of overrunning the system with all these electrons at one time. So one bond at a time means that you have this sequential, keep using the same words, release, loss of electrons. So at that point, here's what you're doing. You have two choices. Pick one. You've got energy in the form of an electron, high energy electron. We can transport it with an electron carrier to the electron transport chain, and we can just squeeze out the energy, use that little bit to do work, squeeze some more out, do some work, squeeze some more energy out, do some work, and then eventually get the electron to where it's given to its final electron acceptor, because we've squeezed all this energy out and it's low enough energy that it can actually be safely given to a final electron acceptor. We can do that, so we get work as we squeeze out the toothpaste of energy, or you have the option, if you want to, of doing it all at once, and you can blow up in heat and light. That's great, because you cannot use all of that energy at one time. So if you release it all at once, you're going to get heat and light. That's it. Or you can sequentially passage it through an electron transport chain, and it's going to get all of those little dabs of toothpaste energy are going to be workable, manageable, usable amounts, and we can do something with it. I kind of like this one over that one. There you go. So let's look at what the pages look like. So I feel like that's, is it zoomed in? Maybe not. No. Okay. So maybe I just need to move me. So we started off up here with our mantra. These are things we're going to say every time we talk about water, behavior, enzymes, or oxygen. The actual notes look like that. And then we had 10 on this page. Maybe we can see it around my head. Do the best.